Good afternoon. So today I am going to talk about eukaryotic gene regulation. In my previous lecture you have seen the prokaryotic gene regulation. So today I have a new topic and go to the first slide. So eukaryotic gene regulation, if you see, we begin from a single cell which undergo mitosis and two daughter cells are produced. In one daughter cell, a special kind of the gene is getting expressed that is known as neuron-specific gene. By the expression of this gene, this cell differentiates into a neuron. While the second cell, it expresses a epithelial-specific gene, and this cell differentiates into a epithelial cell. Now we have a figure up here, bone marrow. You see here a bone marrow section is shown. In the wall of the bone marrow, in between this, you can see a cluster of stem cells. So these cells, they are known as the progenitor stem cell. They can differentiate into multiple type of the cell depending upon the type of gene expression. One is the hematopoietic cell, stem cell. It differentiates into different kinds of the blood cells like the WBC, RBC, lymphocyte, T lymphocyte, B lymphocyte, etc. Another one differentiate into adipocytes, osteoblasts, such cells are produced from the single population of the stem cell. So each cells express a different set of genes, and by the expression of different set of genes, they are able to differentiate into a specific type of cell which perform a distinct function. So every cell in a multicellular eukaryote do not express all its genes. All cells express at only given time only 3 to 5% of all the genes. And when these genes undergo a long term of control of gene expression in the tissue, they differentiate into a particular type of cell that is known as differentiation. And how the regulation is done in the gene expression, to major type of the two kinds, there are two different steps how the genes are regulated in eukaryote. First is regulation at the level of transcription. Second is the regulation at the level of after transcription, that is translation, post-translation modification, other things. The first step is at transcription. And further, if you see, not only at transcription level, there are other steps also, whether transcription will happen or not. So as you see here, there have been shown four different steps. One, remodeling of the chromatin, also known as chromatin modification, chromatin remodeling, transcriptional control, and then processing control, the RNA processing control. And once these mRNA are synthesized, then another transport control. Once it is in the cell, cytoplasm of the cell mRNA, it undergoes another control, it is known as stability control. Once stability control is maintained, then the mRNA proceed for the translation, and this another step of the check controller happens at the level of the translation. And once translation has happened, the primary protein has been synthesized. There is another step of the regulation that is known as post-translational control of protein function. In this case, the folding of the protein is measured, and if the correct folding is not achieved by the protein, it will be the inactive protein. So there are about first step, chromatin remodeling, transcription control, processing control inside the nucleus. 1, 2, 3 steps are in the nucleus, and the 4 transport control, mRNA stability control, translational control, and 7th folding and post-translational modification control. They are outside of the nucleus into the cytoplasm. So we will first study the chromatin regulation. What it does? What is chromatin regulation? As we already know, the chromosome is made up of chromatin, and chromatin is made up of DNA and protein. And proteins are the two types, basic protein and other associated protein. The in chromatin, the DNA is coiled around the histone, and these histones are made up of the nucleosome. Yeah, and this nucleosome unit of the subunit of the histone allows DNA to be packed in the nucleus, but also physically regulate expression of the gene, making them available or not for the gene expression. So the DNA, it is the packaging of the DNA in the nucleus which makes it physically available or unavailable for the gene transcription, and it is regulated at this level. How it is regulated we will see in the next slide. So as you see, so these are the nucleosome particles, and the DNA is wound around this nucleoside particle into this structure, and protein, remodeling protein is an enzyme which binds with these ones, and remodeling protein remove the nucleosome molecule from this and makes a single strand of the DNA. A remodeling protein binds and disaggregates the nucleosome. Now this strand is available for the transcription process. Then the transcription complex binds and mRNA synthesis occurs, and remodeling proteins remains here. They are known as single strand binding protein also. Now the transcription of the complex can lead to the big transcription. And then elongation will be also coming on, and remodeling protein will again come to the next nucleus and modify this one, and by in this way the RNA will be proceeding in the direction the nucleosome will be getting removed. So the second one, chromatin regulation can be of two types, small scale or large scale. In the small scale regulation, the only few part of the chromosome is getting condensed or packed tightly. That is known as heterochromatin. And the other part where the nucleosomes are naked are in that 11 nanometer, 30 nanometer fiber, they are called expressed condition or eukaryotic or relaxed condition. So example of the whole chromosome regulation, that is bar body. That means the bar bodies are the inactivated X chromosome which have undergone total heterochromatization and making it overall inactive. So the small scale means that some part of the chromosomes are inactive, that is known as heterochromatic, and the other parts are the euchromatic. The example of the whole large scale chromosome regulation, that is bar body. So the second one is gene amplification. What is this gene amplification? Suppose a gene is the quantity of a gene's product is required in high amount to increase the gene expression. Make temporary copy of gene occur gene amplification. The genes many temporary copies are produced at the same time. Okay, so how it happens in the tissue, in certain tissue or stage of the development. Embryo require massive volume of ribosomal RNA to make ribosome. In early developmental, there are millions plus extra RNA. Okay, wait for that. So to increase gene expression, the number of temporary copies of the genes are made. It happens in the subtissue like the embryo, where the high number of the ribosomal RNAs are required to make ribosome, and there are millions of extra copy of ribosomal genes not able to replicate or are present in the nucleus. So the transcriptional regulation can occur as you have seen in the prokaryote. The promoter is recognized by a sequence by the RNA polymerase that is known as data box, and RNA polymerase attaches to it and causes the transcription of the gene. And in eukaryote, the promoter consists of a data box sequence and binding of the transcription factor, and a set of sequence that is known as recognition sequence, and RNA polymerase brings about it. So as you see in this figure, this is the lactose like opera model has been shown. As you see in this one, this is the inducer region, Then we have the P lactase, promoter of the lactose overall, and then we have a operator sequence, and here we have a ZYA, the structural genes, and it is the active repressor. When this repressor is bound to the operator sequence, then RNA polymerase cannot bind to the promoter area and cannot transcribe the gene. As you see, it is the repressor regulatory area which controls the transcription of the gene in case of the prokaryote. And when lactose is present, lactose will bind to this repressor, inactivate this one, and it will not be able to bind to the operator area. And now the RNA polymerase will bind to the promoter area and transcribe the gene. But in case of eukaryote, as you see, there is also a tota box, and promoter area is very quite large. There is a initiation start site of the transcription. It binds a set of multiple transcription factors in the area of the promoter, like the TFI transcription factor, second D, A, B, C, D, and these make a large subunit complex, huge complex, along with RNA polymerase to proceed further synthesis of the new mRNA. So, in eukaryote, multiple transcription factor or proteins are required for a gene to get expressed. While in case of the prokaryote, only a few proteins, regulatory proteins, are required to control or to express a particular set of the gene. So, in eukaryote, RNA polymerase interact with promoter, regulator sequence, enhancer sequence to begin transcription, and regulator protein bind to the regulator sequences to activate transcription. Enhancer sequences bind activator protein. Silencer sequences stop transcription. So in eukaryote, the regulatory area consists of many sequences that is known as enhancer sequence, regulator sequence, and also the silencer sequence, which stops and regulator and enhancer is increase the transcription of the gene. Now let us see this one. This is the gene, a particular eukaryotic gene in case of the eukaryotes. So this is the promoter area, and this area denotes the binding of the RNA polymerase binding site, and this is the transcribed area. So it has a transcription factor binding site, regulator protein binding site, and very far from the gene there is another relative sequence that is known as enhancer sequence. It also affects the overall expression of the transcript. So as you see, activator protein binds to the enhancer area, regulator protein bind to the regulatory area, and RNA polymerase along with multiple set of the transcription factor bind to the promoter and RNA polymerase binding site. Once it is bound to this site, the activator protein along with the enhancer sequence loops back and bind to the set of the transcription factor, and binding up all these ones. The DNA creates a DNA bend, the DNA chromatin fiber, and this bend, this loop formation. See how it is makes a long stretch of DNA lies between the activator binding site and the transcription ground. And DNA bending brings the activator protein bound to the enhancer element far from the promoter into contact with the transcription factor. And once the transcription factor, enhancer, and activator proteins are bound, then the transcription goes on. So the expression in eukaryote is quite complex. So what you will see here in eukaryotic genes are typically alone. How to regulate expression of several genes? It means that because in eukaryote genes are not in a set, as we have seen the lactose operon, there are three genes required for the lactose metabolism, they are present together. But in case of eukaryote, each gene exists as individual identity. It has its own promoter, its own regulatory. So how it happens? So see here, there is a mRNA is synthesized from this area. This is a regulator gene, and we have DNA, this is the promoter area, and RNA polymerase binds here and synthesizes the gene. This RNA polymerase can be affected by a external signal that such as an instruction, drought, activate transcription of a regulator protein. And this regulator protein, the master regulator protein, may act as to control a the different gene which may be involved in the same process. See, this is the regulatory protein getting synthesized here. The regulatory protein, and this regulatory protein is like the SRE, stress response element. It may be present at the nearby area of the promoter of the many multiple of the gene, and these regulatory protein bind to this and activate the expression of the different gene at the same time. So eukaryotic genes are typically alone, and but their expression is controlled by a single master regulator protein, and master regulator protein has a recognizing sequence in each of the regulated genes above the promoter area. So once upon we have finished this one, you can explain why gene expression control necessary in eukaryotic cell and describe how gene expression is regulated in before and after transcription. Tell me what is the differentiation in between euchromatin and histochromatin. What is a silencer sequence? And we can explain the gene regulation mechanism different, how it is different in eukaryote and prokaryote. So, first, second one part is that post-transcriptional regulation, how it happens. Okay. So once the mRNA is transcribed in the nucleus of the eukaryotic cell, it undergoes a set of reaction that is known as post-transcriptional regulation. Okay. So first one is that alternative splicing means because as we know the mRNA produced from the transcription is known as nascent mRNA. It has multiple set of sequence in the tandem, that is intron as well as exon, and the change of these are cut alternatively to produce particular mRNA. And whether the mRNA life will be long or short. So produce microRNA that will damage mRNA. So microRNAs are produced in the nucleus and they will bind to the nascent mRNA and degrade it or it prevent it from getting transcripted. Again, then we have guide RNA. Okay, and the RNA can be encrypted to, and the polypeptide sequence produced from that mRNA can also be modified. Okay, so the alternative splicing and the lifespan of the mRNA and the editing of the RNA by the RNA gene editing method. These are the different methods we can further regulate the mRNA and hence the gene expression. Further is the translation regulation. mRNA once present in the cytosol, it is not necessarily that it will undergo further translation. Okay, its rate can be regulated, control the rate of translation. It is the translation can be controlled by the controlling the rate of the translation and gene expression can be held. How? The 5' cap of the processed mRNA, the mature mRNA is modified, and also the feedback regulation is done, and we will see how that. So modify the lifespan of the protein. And the another one that the protein which has been once produced after the translation, if you see a single cellular polypeptide chain, it attaches with a protein that is known as ubiquitin. Once attaches with the ubiquitin protein, it goes to the proteasome complex, it undergoes degradation. Means if the ubiquitin is attached to the any newly synthesized protein, it will not proceed further, it will not exist in the cytosol under the, it will, rather it will go to the proteasome complex and will get degraded into individual amines. So ubiquitination ultimately decides the fate of the translated proteins. So, what are the ways that the cell can regulate gene expression after transcription? How can the process of RNA splicing go on? One pre-mRNA to produce 5 different proteins in the 5 different tissues. We will see now. So can you actually fill this table? What is this? Genome size, a comparison of the prokaryotic. How much is the genome size? How much is there? So basically we know that the genome size of the prokaryotes are in million base pair, while in case of the eukaryote it is trillion base pair. Repeat sequences are present? No, there are no repeat sequences. If it is there, very little will be there. Those are the restriction individualized cut sequences. Or as you have seen the cluster, CRISPR Cas9. Okay, what you say, PIME sequences are there in the genome of the bacteria. And we have multiple replicating sequence in the eukaryotic genome, that is known as long interspersed nucleotide repeat, small interspersed nucleotide repeat. Okay, many repeats are non-coding DNA bit coding sequences. No, they are little. While in case of the non-coding DNA bit coding sequences are present in case of the eukaryote. The transcription and translation is separated in the cell. No. In case of the prokaryote, the translation and transcription happens in the cytoplasm. While in case of the eukaryote, the transcription happens in the nucleus and the translation happens in the cytosol. The DNA is found open to the cytoplasm, which is separated from the cytoplasm in the nucleus. DNA is bound to protein. Yes, it is bound some basic protein, but not the histone proteins. But in case of this one, the basic protein histone binds with the DNA molecule. Promoters are there in the gene, but promoters are flanked by the operator and regulator regions. Here is also promoters are there in the gene, but promoters also have a long regulator sequence upstream as well as downstream of the promoter area. Enhancer and silencers, they are absent in prokaryote. In case of the eukaryote, enhancer and silencers are present. Capping and tailing of the mRNA not required, as the mRNA getting synthesized from the DNA is transcribed, it immediately undergo translation. So there is no need of the capping. While in case of the eukaryote, the capping and tailing of the mRNA is required for the stability of the mRNA. RNA splicing, spliceosome, they are absent in prokaryote. They are present in the eukaryote and do alternative splicing to produce variable gene expression. Number of chromosomes in the genome, usually 1 or 2. Was duplicated. Here the number of chromosomes may vary from 2 to 1200. So this is all about the basics of the eukaryotic gene regulation. In the next presentation, I will discuss in much detail about the regulation of the gene expression in eukaryote step by step, and that will be taken very lengthy and will be okay for the PG student. For UG student, this much is enough. Thank you.